Now, we know that the T-cells are biosynthesized by the precursor cells, which are called pre-thymocytes. And as we discussed earlier, these precursor cells are actually derived by the lymphoid lineage cells, that are ultimately derived by hematopoietic stem cells. And these pre-thymocytes are produced within the bone marrow. And before birth, in fetal liver as well. Once they are produced, then these precursor cells of lymphoid lineage need to migrate towards their site of maturation. That are surrounded lymphoid organs or lymph nodes for b-lymphocytes. And thymus for T-lymphocytes. And we are here talking about T-lymphocytes only. So, let's have a brief look at its maturation site first, that is the thymus. During the embryo developmental stages, the thymus, which is an important lymphatic organ of our body, arises from the third branchial pouch. Thymus is a pyramid-shaped, primary lymphoid organ, and is located in the upper chest, behind the sternum. If we look at its front view, we see a bilobed structure within a mediastinum. Now if we cut it open, we see that the thymus is organized into three major areas, that are the subcapsular zone, which is the outermost portion near the boundary made of epithelial cell layer. The cortex, which lies in between the subcapsular zone and the inner medulla. And the third zone is medulla, which is the innermost zone or portion of the thymus. Different populations of stromal cells and thymocytes are found in each of these structurally and functionally distinct regions. Thymus is functional in young ages, till the age of adolescence. But later, as person ages, it becomes filled with fibrous strands and so it becomes non-functional. So, at young ages, T-cell development is dependent on thymus. But in later stages of life, its range is maintained by the continuous division of already existing T-lymphocytes, and is not affected by the presence or absence of thymus. So, here we briefly talked about the thymus. That is the site of maturation of T-lymphocytes. And within separate parts of thymus, thymocyte development takes place over several stages. And this development is actually marked by the presence of surface markers or surface receptors that are called TCRs or T-cell receptors. And the known receptors that are found in abundance on T-lymphocytes and give rise to major subsets of T-cells are CD4+ and CD8+ surface molecules. So, when pre-thymocytes migrate from bone marrow or fetal liver to the thymus, at this stage they do not express any surface marker. And so they said to be double negative. Here I am writing it as DN. And why they called double negative? Because they are negative for both CD4+ and CD8+ costimulatory molecules. That's why they are referred as double negative cells. At this stage, the T-cell receptors genes are in unarranged configuration. So, these double negative, DN, thymocyte subpopulation then undergoes several important differentiation events. As these DN cells become committed to the T-cell lineage, because TCR gene rearrangements begin to occur. These TCRs or T-cell receptors are heterodimeric membrane proteins, and they resembles in structure with immunoglobulins or antibodies. These are also heterodimers, that means they are made of two different type of chains, and so on that basis, two types of TCRs are commonly found. One type containing alpha-beta chains, found on approximately 95% of human T-lymphocytes. And the other one, containing gamma-delta chains, present on approximately 5% of human T-lymphocytes. So, here at this double negative stage, cells make important choice where they get committed to either or T-cell lineage. Notably, both these T-cell lineages derive from a common progenitor cell, with commitment to one lineage or the other being a competitive process based on which TCR genes productively rearrange first. And ultimately, the majority of thymocytes commit to the alpha-beta TCR pathway, and only a small number of thymocytes commit to the gamma-delta pathway. Because gamma-delta T cells are not MHC restricted, they are not subject to the other thymic developmental processes and leave the thymus at this stage. Let me try to simply explain this process of TCR gene rearrangements. Well, at this DN stage, rearrangement of TCR genes begin, where the beta, gamma, and delta genes all attempt to rearrange at this stage. And this rearrangement involves activation of recombination activating genes, RAGs, and terminal deoxynucleotransferase, TDT. This TDT contributes to the production of a variety of antigen receptors over the surface of T cells. And these recombinases, that are produced by the immature T cells, under the influence of thymic hormones, are mainly IgG1 and IgG2. They shuffle the DNA to give rise to particular kind of TCRs according to the antigen present. We will not talk in detail about the gene rearrangement of T cell receptors here, because you will get another complete video on TCRs in detail very soon, so just wait for it. But for this video, it's enough to know that at this stage, under the influence of thymic hormones, many enzymes are released by immature T cells, or pre-thymocytes, such as TDT and recombinases. These enzymes play a role in DNA shuffling and production of different kind of surface markers or TCRs. Here, I have mentioned thymic hormones. These hormones are actually released by the epithelial cells of thymus in the subcapsular zone. These hormones mainly include thymulin, thymosin, and thymopoietin, etc. These hormones act as chemokines for the attraction of pre-thymocytes from bone marrow to the thymus. And also, at this stage they stimulate the production of enzymes. These enzymes then help in producing antigen receptors. Also they activate genes, giving rise to CD, that are cluster differentiation proteins. After all the gene rearrangements, either alpha-beta TCR lineage is formed, or the lineage is established, along with CD proteins. All these process takes place within the subcapsular zone. After that, the next stage starts in the T-cell development pathway, and that stage is the double positive, or DP, stage, due to the presence of double positive cells. These cells are called double positive, because now, under the influence of recombinases, both cluster differentiation proteins that are CD4+, and CD8+, are found over the surface of now called thymocytes. And these thymocytes are abundantly present in the cortex of the thymus. And these are double positive, that means, although CD surface markers are now present on their surface, but they haven't decided their fate yet. Means, they haven't selected their lineage, if they want to be CD4+ or CD8+ T cells. So, these DP cells express both CD4 and CD8, and such cells are not found outside the thymic cortex. Till this stage, thymocytes have developed either alpha-beta or gamma-delta TCRs, along with both cluster differentiation or CD proteins. Now, from here, the positive and negative selection of T lymphocytes occurs within the medullary zone of the thymus.
Watch our medical videos anytime and anywhere. Download sqadia.com app now.